All right, so our next speaker is Anastasia Hains. So Anastasia did her undergrad at St. Olaf College, followed by master's and PhD degrees at the University of Chicago. Her PhD was with Lucy Godley, focusing on regulation of 5-hydroxymethylcytosine and pediatric neuroblastoma. She then moved to Stanford for her postdoc in the Van Renchim lab, where she has been studying epigenetic regulation of DPIG over the cell cycle. And I'm very excited for her talk today on diffuse midline gliomas and present altered DNA replication and are susceptible to replication stress therapy. So, Anastasia, over to you.
Thank you, Ben. And I am going to be sticking with H3K27 as well. However, I will be presenting about an entirely different context. So diffuse midline gliomas, also known as diffuse intrinsic pontine gliomas, or DIPG, are pediatric brain tumors that arise in the pons region of the brain, which makes surgical intervention impossible. And therefore, this disease is uniformly fatal. The tumor arises usually upon with the first mutation of histone H3K27-M, which is present in greater than 80% of all DMG tumors. So this mutation can actually arise in one of two genes, the H3.1, which is the replicative canonical histone protein, and H3.3, which is the variant. Although very similar, the two proteins actually have important distinctions. They differ on the basis of 5 amino acids, PTMs, different chaperone proteins, and genomic localization. But for my work, it's important to highlight that H3.1 is only expressed during S phase, and H3.3 is expressed throughout the cell cycle. In normal cells, when H3K27 is wild type, this mark is usually modified with either an acetyl group, which was presented in Anna's talk, or to a trimethylation mark, which is generated by the protein EZH2, part of the PRC2 complex. This is known generally as a repressive mark and then ensures that transcription regulation is turned off. However, when the H3K27M mutant is introduced into the genome, not only is there loss of any modification going on at the site of the mutation, it actually acts as an inhibitor of EZH2's activity and therefore prevents the spread of trimethylation at the K27, at the wild type K27s. This also leads to a global increase in acetylation of H3K27, leading to aberrant transcriptional activation, a reduction of trimethylation, leading to aberrant repression of genes, and then spikes of trimethylation at other chromatin loci results in aberrant decreased gene expression. And as you can tell from the system, this mutation is not easily targetable, leaving us to search for downstream targets as a potential therapeutic. Now, during my advisor's postdoc, she actually found that H3K27 trimethylation associates with specific replication features. What you're looking at here is replication timing sequencing, also called RepliSeq, where BRDU is incubated with cells in culture for a short period of time, so it can be taken up in the nascent replicating strands of DNA. The cells are then FACS sorted based on DNA content, so then we can divide the S phase into 4 phases. The cells are then, taken back and processed, and BRDU is immunoprecipitated, and the strands are then sequenced, generating these figures when all of the different phases are put together. And you can easily see their distinguishable replication timing features, initiation zones, termination sites, termination constant timing regions, and initiation constant timing regions. And when you compare this uh, uh, global sequencing with other big data sequencing results, you find that termination sites were in particular well represented with K27 trimethylation. And in particular, when Dr. Van Reckum looked at enriched regions of trimethylation, she found that these regions broke down into fairly proportional early, mid, and late replicators. Um, but when looking at when the mark was enriched, the early replicators were particularly enriched during G1 and G2. The mid-replicating regions were enriched in G1, early S, and G2, and late replication regions were enriched in early S or late S, meaning it's important to note that this trimethylation mark was absent when replication was actively ongoing. And therefore leading us to ask the question, what happens in a system like DMG where there's global loss in this trimethylation mark? How does replication timing and cell expression, gene expression change? So to answer this, you decided to use 6 DMG cell lines, 3 H3.1 K27M and 3 H3.3 K27M, in order to acknowledge the distinction of when H3.1 is only expressed during S phase, and then 2 astrocyte cell lines as a control, as astrocytes are abundant normal cell in the brain, and they are also fairly actively replicating. For our RepliSeq experiments, we cell sorted the cells based on DNA content and broke S phase down into 4 phases, just as I previously showed. And for RNA sequencing, we divided the entire cell cycle, G1, early S phase, late S phase, and G2. And when we looked at our replication timing results, sure enough, we found that each of the cell types had unique replication timing patterns. Here I'm just showing an example, but when we looked and analyzed globally, we found that both H3.1 and H3.3 K27M, when compared to astrocytes, had about 10% of the genome shifted to differential replication timing. However, H3.1 K27M, when we looked specifically over whether this shift was earlier versus later, we found more than half of the shifted regions were later. And when this was broken down into the specific replication timing features I mentioned earlier, initiation zones were particularly abundant, but this earlier and later change was pretty much divided proportionally amongst these replication timing features. H3.3 K27M, on the other hand, instead had over half of the differential regions replicating earlier. And most of these were coming out of the termination sites. So our hypothesis that there may be differences between H3.1 K27M and H3.3 K27M was so far bearing out. And it followed up with our additional question of, are there any correlation between these differential replication timing regions as well as gene expression? And we found this was true as well. What you're looking at is a correlation between replication timing index and differential gene expression. So you can see as regions shift later or earlier, gene expression is also changing in correlation. So the regions of the genome that shift later have a proportionally a lower fold change in gene expression, and vice versa. This was also true when we compared each of the DMG phenotypes with astrocytes, as well as when we compared H3.3 K27M and H3.1 K27M together. So what were these genes that were being differentially expressed? Well, we broke down the analysis of these differentially expressed genes, in particular based on our sorting method. We found when genes were differentially expressed, predominantly all that differential expression was shared between all cell phases. But looking at unique cell phases, S phase was the dominant a phase that contained genes that were differentially replicated or differentially expressed. And when we performed gene ontology analysis on these genes that were differentially expressed in S phase, we were pleased to find that most of these genes were highly involved in DNA replication. And what's more, not only were these genes differentially expressed, their timing of express was much different than astrocytes as well. So if you see here, we're just kind of going through cell cycle, G1, early S, late S, G2, and back to G1 again. But where astrocytes have a sharp increase in these DNA replication genes between early S and late S, the DMG cell lines are upregulating earlier between G1 and early S phase. This led us to hypothesize that not just gene expression was being changed, but the replication fork machinery was also being altered in these DMG cell lines. So we collaborated with the Torres Padilla lab, and they performed the DNA Farber assays to look at replication forks in these cells. And we found that interorigin distances were significantly longer in the astrocyte cell lines, indicating that there are actually more origins in DMG cell lines. And simultaneously, replication fork speed was much faster in the astrocyte cell lines than the DMG cell lines. These can be indications of replication stress. So we looked for signs of replication stress in our DMG cell lines, and we indeed found out that the gamma H2AX, which is a sign of DNA damage, is present in the DMG cell lines, but not in the astrocytes. And this is just at baseline. We also turned to our collaborators again, this time the Philbin lab, who had done massive single-cell RNA-seq experiments with K27M tumors. We asked them to look for replication stress signature in these cells, as well as normal hippocampus cells, and found that the replication stress signature was significantly higher in the K27M mutated cell lines compared to normal cell lines, indicating that DMG tumors and cell lines are at replication stress at baseline. So what happened when we treated these cell lines, as well as our control astrocytes, with replication stress therapeutics? I started with hydroxyurea, and we found that the astrocytes were significantly more resistant to treatment with hydroxyurea, and the DMG cell lines were significantly more sensitive. But this is just one therapeutic, and I wanted to test for more, as well as our reviewers wanted to see additional replication stress therapeutics. So I also tested fludarabine and berzosertib and found that the pattern held true, in which case the DMG cell lines were significantly more sensitive to replication stress therapeutics than the astrocytes. This work is promising because these replication stress therapeutics, all that I tested, are able to pass the blood-brain barrier, which is a significant challenge when it comes to treating these tumors. And we are currently in the process of testing in vivo models. And so far the results, which I'm not going to show today, but the results look very promising. And with that, I'd like to finish up by thanking my mentor in my lab, as well as our collaborators in the Philbin, Torres Padilla, and the Sed Dureyev laboratory, in addition with my funding sources. And of course, if you would like to see more information, this paper was published back in December, and you can find it online.
Thank you. That was fantastic. I really enjoyed that. So reminder, if you have questions, you can type them into the Q&A or click the raise hand button to ask them yourself, but I'll start off. So um, did you look in these K27M, like I know with other histone modifications, like with K9 methylation and demethylases, that sometimes been linked with like over-replication? Um, have you looked at that with the K27M, if that's the phenotype you see?
So no, it's a short answer to answer but yeah, so we are in the process of,
Mhm,
so it's hard to look at and draw conclusions from like cause and effects with the cell
mhm,
lines that we have. So we are actually in the process of, we have inducible K27M cell lines. And we're right now, like, we're literally testing antibodies against a whole slew of different histone marks, to basically do exactly as you said. So it's happening, but no, like other histone marks we haven't looked yet.
yeah. And do you see like, um, you kind of have the gamma H2AX signal, and you've
Mhm.
got this difference in replication speed. Do you see particular types of DNA damage being induced or different repair pathways that these are sensitive to or dependent on?
Yeah, that's a great question. I haven't tried. Um, I haven't really looked either. I briefly looked for like check one, and phospho check one. But essentially it came down to those antibodies weren't working and we really wanted to
Mhm.
send the paper in.
Yeah.
So but yeah, it would be I think a great, we have a whole list of drugs we would love to test on these, and PARP inhibitors are
Mhm.
definitely on that list of drugs to test. But like I just haven't gotten there yet. But yeah, that's a great idea.
Yeah. And you could even envision like potentially some of the replication stress drugs, like those drugs in combination with something like a PARP inhibitor, maybe
Oh
they're
yeah. Oh yeah.
sensitized to it.
Especially since right now standard of care is to treat these patients with radiation. So in like our perfect world, we would have our chemotherapy combination as well as the radiation
Yeah.
therapy. Mhm.
Yeah. No, it's a really cool framework to think about how the
Mhm.
different therapeutics may come together. So the question, but I'll just ask one question out of the chat, asking, have you looked at the amounts of the K27M um, for H3.3 and H3.2 in the genome, and if they make up similar percentage of total H3 of the genome in these cell lines?
Um, Not I in particular no, but I know that generally there is no compensation with this disease. There really is like a total loss because once you get the knockout version of these cells, the Jabato lab, I think they were the first, but now a bunch of labs have followed suit. They actually generated DIPG cell lines that had the mutation knocked out, meaning they're reverted to wild type,
Mm,
and there was a global gain in the trimethylation mark, and the cells were decently recovered. And in particular with the trimethylation, I'm not sure about H3.2 though.
yeah. Great. Okay, so we'll wrap up there. There's another question or two in the chat, so you can go ahead and answer those by typing. But thank you for the fantastic talk.